Good morning, everybody. The Center for Integrative Environmental Health Sciences and the Department of Pharmacology and Toxicology welcomes you to our Environmental Health Sciences seminar. I would like to introduce Dr. Mayukh Banerjee. Dr. Banerjee initially received his bachelor's degree in zoology in India and his doctoral degree in 2010 from the Institute of Chemical Biology in Kolkata, India. His graduate project involves studying genetic susceptibility to skin cancer development in an arsenic-exposed population. After graduating, he moved to the University of Alberta in Canada, where he performed postdoctoral research studying cellular arsenic transport, completely funded by the Alberta Cancer Foundation, excuse me. In 2018, Dr. Banerjee joined the Pharm Tox department here at U of L, working under the mentorship of Dr. States. And in 2020, he was recruited by the department as its first P30 CIEHS Career Development Awardee. And his current research focuses on how population-relevant environmental arsenic exposure causes multi-organ cancer and its mitigation. Dr. Banerjee has co-authored 42 peer-reviewed publications and 4 book chapters. He's been invited to and presented at multiple international, national, and regional conferences. In 2022, Dr. Banerjee was recognized by the Society of Toxicology with the Paper of the Year Award, specializing in dermal toxicology, and the Young Investigator Award by the O-Value chapter. His current research is funded by the American Cancer Society and the NIH. And as we all know, he teaches tox courses here and directs our seminar program. So a fun fact about Dr. Banerjee is that he loves bird watching and bird photography. And he actually has a first author publication documenting the first photo record of a bird called fieldfare from the Indian subcontinent. So without further ado, Dr. Banerjee.
Thanks, Dakota, for that kind introduction. Good morning to all of you. So my talk will try to explain how arsenic and zinc finger proteins interact with each other and how that modulates multi-organ carcinogenesis. So Dakota has given a brief introduction of what I have done, but I will provide a little bit more context by discussing why it is important to study arsenic and what is already known, which will lead me to what are the questions that still need to be answered. I will show you some data from my selected published work, which informs my long-term research goals. I will show you preliminary data, mostly unpublished, that I have been generating for extramural grant fundings. I will also show you some data from my ongoing currently funded research work from American Cancer Society and wrap up with conclusions, future directions, and acknowledgements. So Dakota has already said this, but obviously I want to beat my own drum. So I started my research career working at the Indian Institute of Chemical Biology, working on the arsenic-exposed population of West Bengal, India, with a brief stint at the University of Manchester in UK. Following that, I moved to Canada because they were climatically very similar to study how arsenic is transported within our body. And then I moved to the University of Louisville, this department, trying to understand the molecular mechanisms of arsenic-induced skin carcinogenesis with Dr. States. And when I was recruited as a faculty, I started focusing on zinc fingers as possible modulators of arsenic-induced carcinogenesis. 230 million people, and possibly more, across 108 different countries are exposed chronically to arsenic from environmental sources, mostly from drinking contaminated water. If you're wondering, why do people drink this contaminated water? It's because it's colorless, odorless, tasteless, and it's very, very difficult to measure it with technologies that are, people have at hand, unless you have an ICP-MS lying around somewhere. That includes 4.1 million people currently exposed in the US, but this number is slated to rise in the coming decades due to climate change related drought. Two things that I want to bring to your focus is, if you see all these white areas in this world map, that is not really safe from arsenic. It's really the areas where we have no data. So as more and more data comes in, we probably will see this 230 million also go up and this 108 also go up. The second thing is, if you look at this very intense red dot right about here, which represents about 120 million exposed people across India and Bangladesh, that's where I grew up. So I have actually experienced personally families and communities falling apart from this insidious exposure, which really adds a layer of what I can do about it, in addition to my intellectual curiosity, which really drives my research. And this exposure gives rise to multi-organ cancers, as established for skin, lung, liver, and bladder, as also late-life cancer risk from in utero exposure. Also causes multi-organ non-cancerous disease outcomes and spans across different developmental stages of the life, also associated with lifestyle-associated disorders. So it pretty much does everything you can think of. Today's talk will be focused mostly on skin cancer, some of which are presented here in this photograph that I took during my graduate studies working with these people, and lung cancer. Now, unfortunately, there is no cure for any of the arsenic-induced diseases. What is recommended is to provide arsenic-free potable water. However, the fiscal investment required to do that is huge. So this is an estimate that was done in 2010. So billions of US dollars are required to provide the exposed population of Bangladesh with potable water for 10 years. And this, I'm sure, has only gone up in here. So while it is possible, I don't really know if it's feasible, especially for a country with a GDP of $1,100 a year. Also, it is not really effective for people who have already been exposed either in utero or early in life. So the bottom line is we need to understand at the molecular level how environmental arsenic exposure causes multi-organ cancers, because this will allow us to identify what are the putative therapeutic targets, and based on that, possible mitigation strategies. The first definitive report of arsenic ingestion is associated with skin cancer was published in 1887. And since then, if you look at PubMed with the words arsenic and cancer, there are almost about 9,500 publications, of which I'm happy to say I contributed to a few. So we do have a wealth of knowledge about what arsenic does. And depending on where you're looking at the literature, it pretty much disrupts any cellular and biochemical process you can think about. Despite all this information, we do not understand how arsenic disrupts any of these processes. And the reason being, we know that arsenic does not bind DNA, yet it disrupts a lot of DNA metabolic processes. It does not bind to RNA, and it does the same with RNA metabolic processes. It also changes protein translation, synthesis, stability, chemical modification, as well as degradation, but we really have very little idea of what it is directly interacting with in cells to give rise to all these effects. And this really comes down to three major caveats that exist in the current state of literature. I'll come to them one by one, but to summarize, first one is most of the mechanistic studies that you will find that if you look into the publication use very high concentration, but very short duration to model very low concentration, very long duration exposure, which is not necessarily appropriate. And I'll come to that why. Secondly, there is a lack of historical animal models to study arsenic-induced carcinogenesis. And at no concentration whatsoever, we have been able to induce skin cancer by arsenic exposure alone so far in any animal model. And thirdly, there is very limited knowledge about what are the direct interacting targets of arsenic in the cells that give rise to all sorts of effects that we see. Now, coming to the first one. So the steady state plasma arsenic concentration in people with no known arsenic exposure is around 20 nanomolar. In people who are exposed environmentally, whether it be at the lower end of the spectrum at 10 parts per billion or at the higher end at 800 parts per billion, the plasma arsenic concentration remains remarkably steady at around 100 nanomolars because we are really good at metabolizing and excreting most of the arsenic. In very rare scenario, when people are treated with direct infusion of arsenic trioxide to treat acute promyelocytic leukemia patients, it can rise as high as 5 micromolar very transiently around 48 hours after the transfusion, but it comes down to basal level very rapidly around 72 hours. So the issue here is most of the studies have been done in and around this therapeutic or even higher levels. And the problem with that is arsenic dose response is very rarely, if ever, linear. As these are two representative bar graphs, which actually shows there is a higher biological response at a much lower concentration, and that goes really away when you treat the cells with the higher exposures. So it is inappropriate to take a high concentration study and extrapolate that to explain what's happening when people are actually exposed to 100 nanomolar. That's what most of the internal cells of our body face on a continuous basis, with the exception of bladder, liver, and placenta. And I really like this figure because it kind of summarizes what are the most well-known and most well-proposed mechanisms that are associated with carcinogenesis and what concentration do we see the effects. And if you see those are mostly around this, which is micromolar to high micromolar levels. Barely anything is going on at this nanomolar level, which we know is the body load of arsenic that people face. Coming to the rodent models. So there are known interspecific differences in how humans and rodents metabolize, sequester, and excrete arsenic. Rodents are usually way better than us, and we are pretty good ourselves. So in the human liver, arsenic is metabolized by a sequence of oxidative methylation followed by reduction, and this goes on for 2 cycles. And we excrete most of the arsenic as the dimethylated pentavalent forms. Rodents do this another step and excrete everything in the trimethylated forms, which are way less toxic than any of these forms. Furthermore, rats can sequester really large quantities of arsenic bound to hemoglobin in their red blood cells, which humans cannot. And thus, in rats, this amount of arsenic is not really available to cause toxic effects to the cell. And all these interspecific differences become very apparent when you look at the animal models that have been used to study arsenic-induced carcinogenicity. If you look at these concentrations, these are parts per million, not parts per billion. So they are 1,000 to 1,000,000 folds higher than what humans are exposed to and what humans have in their body. And this amount of arsenic is required to induce a slight increase in the number of tumors in rodent models that are already genetically predisposed to form tumors, as well as in association with another standalone carcinogen such as UV. So that creates a lot of confounding to try and extrapolate that result to explain what's happening in people. Furthermore, if humans were exposed to any of these concentrations of arsenic, they would die within the hours to days just because their gut has been completely obliterated. It will not cause cancer. And thirdly, again, as I said before, we don't really know what are the arsenic interacting targets in the cell that mediates any of its effects. We do know, though, that arsenite, inorganic arsenite, has a very strong affinity for sulfhydryl groups that are found in free thiols. We are enriched in amino acid cysteine, for example. In fact, interaction with protein sulfhydryls was proposed to be a possible mode of arsenic carcinogenicity back in 1998 by IR, although empirical data remained very elusive, at least till 2010. And these cysteines are really enriched in zinc finger proteins, which make them possible targets of arsenic toxicity. And in the last 15 years, our group and others have started showing that arsenite can actually bind to these zinc fingers that are coordinated into the proteins either by 2 cysteines, 2 histidines, 3 cysteines, 1 histidine, or 4 cysteines. So arsenite can only displace those with 3 cysteines, 1 histidine, or 4 cysteines, but not from the ones that are 2 cysteines and 2 histidines. However, the issue is there are around 950 different proteins that has at least one of these C3H1 or C4, and we have only studied 15. And again, look at the concentration at which these are studied. So you're studying something at 20 micromolar. This may not just happen when multiple zinc fingers in the cell are competing for available limiting 100 nanomolar arsenite concentration. So we really, really need to do the studies that are population relevant concentrations. So based on all of this, there were some key knowledge gaps that we really have to answer if we want to get an understanding of how arsenic causes cancer. So the first one is what cellular processes are actually disrupted by arsenic at the population relevant 100 nanomolar exposure. Secondly, which arsenic targets mediate the effects of arsenic by disrupting these processes? And thirdly, sorry, how do these disruptions turn healthy cells cancerous? So when I started working with Dr. States, we made use of a model that was developed by Pai et al. At NIEHS, where they took uh, target human skin cell, and these are HECAT cells, and these are spontaneously immortalized but non-tumorigenic cells. And if you expose them continuously for 28 weeks at population relevant 100 nanomolar arsenite, they give rise to fully transformed cutaneous squamous cell carcinoma phenotype, which is what we see in patients who are actually exposed. We had independent quadruplicates in each condition to account for biological variability. We also had passage matched controls at each time point, and this is very important to rule out or figure out what are the effects of passaging time, because we are culturing cells for so long, and what are the effects of arsenic at each time point. And using this model system, what we did was we did multi-omics analysis at 3 key points during the development of cancer. So we did both transcriptomics, differential alternative splicing, as well as proteomics. And the 3 key time points are 7 weeks, which are the early adaptive changes that the cells are trying to do when they are faced with the arsenic challenge to adapt to that. By 19 weeks, we see the first signs of transformation, but by no means the cells are fully transformed. However, at 28 weeks, they have transformed completely and are showing the cutaneous squamous cell carcinoma phenotype. So with all of these experiments, what we saw was whether we are doing differential gene expression, differential splicing, or differential proteomics, at each time point, there were hundreds to thousands of genes or proteins that were differentially expressed or spliced. The take-home message here is these changes are temporally dynamic. So if you see here or here or here, the overlaps between the changes at the different time points are little, whereas most of them are unique. So in other words, one set of changes lead to the next set of changes, lead to the next set of changes, and they almost like a wave function give rise to carcinogenesis, which is what we also saw when we used this individual gene or protein level data to do gene ontology or IPA pathway analysis. Again, this is a lot of data, but the take-home message is if we look at what are the changes in the cells that are transformed, that does not tell us what changes actually drove the initiation and progression of transformation. We have to look earlier. So based on all this data, we were able to distill it down into a few key biological processes that were disrupted by 100 nanomolar arsenite exposure before the cells were fully transformed. And we think these are the processes that contribute to arsenic-induced carcinogenesis. And when we started looking at the literature, we were able to show that, well, we were able to see that a lot of these can be explained by the interaction of arsenic with zinc finger proteins that apically regulate each of these processes. So people have shown that arsenic is capable of displacing the zinc from these zinc finger proteins, which in turn disrupts the processes of the host protein that regulate these processes. When I started looking into the other 4, we quickly found that these were also apically regulated by a host of zinc finger proteins that are likely targets of arsenic toxicity and arsenic-induced zinc displacement. Furthermore, literature suggested that all of these processes require zinc for proper smooth functioning. And if there is dietary zinc insufficiency, especially if that is coupled to arsenic exposure, that will exacerbate the arsenic's disruptive effect on these processes and even make people more prone to develop skin lesions as well as skin cancer. So we were able to hypothesize that the interaction of zinc fingers, and arsenic is probably a likely contributing factor in how cells become cancerous. So when I started my tenure as a faculty, I had 3 major long-term research questions that I really want to solve. The first one is, what are the potential arsenic targets that mediate these observed cellular effects? Secondly, how do the interaction between arsenic and its cognate target zinc finger protein cause the disruption that we have already observed? And thirdly, if this is being driven by arsenic binding to and displacing zinc from these zinc fingers, can we put in more zinc and prevent that? So today I am going to talk mostly about these two processes, the alternative splicing and the RNA M6A methylation in the context of arsenic toxicity and cancer. So I wanted to understand how arsenic is disrupting alternative splicing, because we have already seen that happen in HECAT cells. Why is alternative splicing important? Well, this is a homeostatic process that is taking place in most cells in our body, where a single primary transcript yields different mature RNA, and in many cases, different protein isoforms by reshuffling of the exons and introns present in a given gene. And this is actually one of the ways to fine-tune the expression of protein isoforms, mRNA isoforms, but also in many cases, it really controls how much of a canonical protein isoform is being produced by minimizing the canonical isoform at the expense of isoforms that do not ever translate to protein. This is important because it occurs in about 94% of all human genes. Also, disruption of alternative splicing is known to drive carcinogenesis, as this figure shows here. Many of the alternative splicing genes that are critical in cancer are known to be spliced differently between cancer tissues and cognate non-cancer tissues. And we have already shown that arsenic disrupts splicing in the skin cell line. So I wanted to answer two major questions here. The first is, does arsenic induce differential alternative splicing in other target tissue as well? Or is this a core feature across different targets? And secondly, if yes, how does arsenic do this? Because it does not bind to any of these RNAs. So to answer the first question, we exposed immortalized human lung epithelial cells that are non-tumorigenic, called BES2Bs, to 100 nanomolar arsenite for 8 weeks. And we were able to show that both, like in skin cell lines, there were thousands of differential splicing events across all the 5 major subtypes of alternative splicing. So yes, it does induce a lot of differential alternative splicing in different target tissues. We were also able to identify a set of 14 genes that were differentially spliced in both the tissues. And differential splicing, as well as expression of all 14 of these, are known to be important in cancer. So we had identified a group of genes that are likely driving carcinogenesis. The question is how? I started looking at how alternative splicing program is regulated in the cells. And we soon came up with this group of proteins called splice regulator protein network. This is composed of 15 proteins, which are all rich in serine and arginine. This consists of 12 members of the SRSF family, SRSF1 to 12. It also contains TRA2A, TRA2B, and 0 and B2. And each of these have hundreds to thousands of targets, genes in the transcriptome that it can modulate the splicing and expression of. However, what is really important and interesting is within this network, each of them really intricately regulates and coordinates the splicing and expression of all the other members. So if the splicing or expression of one of these nodes is affected, that cascades down this entire network. So I started looking at if there are any arsenic targets in this, and what we found was, yes, SRSF7 has a C3H1 zinc finger, and 0NB2 has 2 zinc fingers, which are C4s, that are likely targets. So we started working on the hypothesis is arsenic is binding to and displacing zinc from 0NB2 and SRSF7, which will then change the splicing and expression of the targets TRA2B, SRSF7 itself, and SRSF3, in turn, cascading down the entire SRSF network. And if the network is dysregulated, so will be the transcriptome-wide differential alternative splicing, which we have already seen. So arsenic does not need to bind to each of the RNAs, but it can bind to a couple of them, and that will explain what we have observed. So in order to understand if arsenic actually binds to any of these zinc fingers, what we did was we synthesized peptides corresponding to the 2 0NB2 zinc fingers and the 1 SRSF zinc fingers, and incubated them with increasing concentrations of arsenic. And we were able to show that all the 3 zinc fingers across the 2 proteins bound arsenic in a dose-dependent manner. We were able to corroborate this by doing direct mass spec studies, where the mass of these peptides increased commensurate with arsenic binding when these peptides were incubated with arsenic. Next, we wanted to know, well, yes, arsenic binds to it, but does arsenic displace zinc, which you would expect these proteins to be normally bound to in normal circumstances. So what we did was we took these peptides, we incubated them with saturating concentrations of zinc, and then incubated them with increasing concentrations of arsenic, and measured if arsenic is binding and zinc is being displaced. And we were able to show that yes, arsenic is still binding in a dose-dependent manner, and it does so by decreasing the zinc binding from each of these peptides. So arsenic is going in, displacing the zinc binding in its place. So next we move on to function. So we take this to cellular systems where we exposed skin cell lines, HECAT or Quercity, and lung cell line PS2B chronically to 100 nanomolar arsenite. And so the function we are looking at is ZRANB2 regulates and minimizes the expression of this beta-1 isoform of one of the other SR proteins, TRA2B. So the beta-1 gives rise to the canonical TRA2B protein isoform. So ZRANB2 minimizes this by promoting the formation of this alternative beta-3 isoform. And we were able to show that this beta-3 isoform, which is ZRANB2 dependent, goes down at the expense of this canonical isoform beta-1 in each cell line. We also corroborated this at the level of proteins, where we saw that TRA2B canonical isoform is up across all 3 cell lines. Now, if TRA2B is more, it affects other members of the SRSF. So as a reporter, we studied SRSF1. So if there's more TRA2B, it will produce more SRSF1, which is what we also saw in all the 3 cell lines. So next, we move on to SRSF7. SRSF7 is a little odd because its function, primary function, is to auto-splice itself to minimize its own protein expression. And what it does so by promoting this SRSF203 isoform that does not give rise to a protein that is degraded by nonsense-mediated decay at the expense of this 201 isoform, which is the protein-giving isoform. So when we expose these cells to arsenic, we actually saw more 201 to 203 ratio across all 3 cell lines. So we expect more SRSF7 protein being made in the cells, which is, again, what we see in all 3 cell lines. Now, although there is more SRSF7, SRSF7 is also a target for arsenic binding, which minimizes its function. So in normal circumstances, SRSF7 will minimize the expression of SRSF3, but there is more SRSF7, but it's not functional. So we expect more SRSF3 as well, which is exactly what we see. At this point, we were wondering, well, yeah, we understand what arsenic is doing, but can we prevent this if we put more zinc in? So we went back to the peptides. In this case, what we did was incubated the peptides with saturating concentrations of arsenic and then incubated them with increasing concentrations of zinc. And we were able to show that, yes, zinc goes in and binds at the expense of arsenic, which is now getting off from the peptides. And we took it back to the cell system. And this work was done with considerable help from my summer student, J.C. Mastic. So we exposed 2 skin and 2 lung cell lines chronically to arsenic, 100 nanomolar arsenite for 48 hours, and concomitantly supplemented them with 1 micromolar zinc. And we showed that, yes, in presence of arsenic, the ZRANB2 dependent beta-3 isoform is down, but that's back to basal level when the cells are supplemented with zinc. We are also able to show the same for SRSF7, where this 201 to 203 ratio was back to basal level for each of the 4 cell lines when the cells were supplemented with zinc in addition to arsenic. So together, we have established that ZRANB2 and SRSF7 are direct arsenic interacting targets that mediates its effect on the splice regulatory network. We have shown that arsenic binding disrupts ZRANB2 and SRSF splicing function that then cascades down throughout the regulatory network and is likely to cascade down to the entire transcriptome, which explains why we see hundreds and thousands of differential alternative splicing events going on at each time point. Now, moving on to the next project, also wanted to understand how arsenic, if and how arsenic disrupts mRNA, M6A mRNA methylation. So RNA is mRNA, RNA in general and mRNA in particular is known to be heavily chemically modified by different processes. We know about more than 100 of these post-synthetic chemical modifications, of which N6-methyl adenosine, or M6A, is the most common and possibly the most widely studied. This regulates gene expression as well as alternative splicing and a host of other RNA metabolic processes. Um, it's known the disruption of normal M6A signature is known to disrupt transcriptome expression and lead to carcinogenesis. And I was interested because this metal 3, which is the catalytic subunit of the writer complex and is responsible for the transfer of the methyl group from the donor SAM onto the target mRNA, has two C3H1 zinc fingers, which are also shown here, that are prime targets for arsenic-induced zinc displacement. In fact, people have shown that if each of these zinc finger domains are mutated so that they cannot bind zinc, that almost completely obliterates the catalytic activity of metal 3. So we were working on the hypothesis that yes, arsenic goes in, binds to this, gets rid of the zinc, that will suppress metal 3 catalytic activity, that will change the methylation pattern on the cells, and that is likely to drive carcinogenesis. So we wanted to answer the question that does environmental arsenic exposure impact M6A levels in the first place? And if it does it do so by binding to metal 3 and disrupting its function? And finally, how does this differential or suppressed metal 3 activity change the epitranscriptomic signature in the cells and contribute to carcinogenesis? So today I'm going to show you some of the data from the first 2 points. And this work was driven by my undergrad student, Madeline Martinez. She standardized a lot of these processes, generated a lot of this data. So I'm really grateful that she helped out with that. So the first set of experiments we conducted was took HECAT cells, exposed them to 100 nanomolar arsenite for 48 hours, and measured the total M6A content. And yes, it went down. So this is total M6A content, not just in mRNA, across all RNA. We are also able to show that, yes, this is zinc dependent, because when you supplement them with zinc, not only it prevents the arsenic mediated suppression, but it actually goes beyond the basal levels. So next, we wanted to see if this is being driven by arsenic's effect on the metal 3 catalytic activity. So we used a commercially available metal 3 activity assay kit and incubated the kit with increasing concentrations of arsenic. And we were able to show that at 2 molar equivalents, which is around 68 nanomolars, there was a 60% loss in metal 3 activity. The next question was, can we add zinc to this system, and will that restore metal 3 catalytic activity? So we did that assay. So we incubated the system first with 2 equivalent or 68 nanomolar arsenite, which we know suppresses 60% of the activity, and then incubated the system with increasing concentrations of zinc. And we showed that, yes, there was a dose-dependent increase in metal 3 activity, even going beyond basal levels at the highest concentration tested, which nicely aligns with our last set of data on the cells. With all this data, we are now primed to look at if arsenic actually binds to metal 3 or not. So we synthesized the wild type C3H1 zinc fingers, and metal 3 has 2 of them. So this is the first one. This is the second one, where each of them have C3H1 motif. We also synthesized corresponding C2H2 mutants, because those are not likely to bind arsenite, but will bind zinc. And again, we did arsenic binding and zinc binding assays, and we were able to show that, yes, in each cases, the wild type, which is in blue, binds arsenic in a dose-dependent manner, but that binding is completely lost if the C3H1 is converted to C2H2s. However, zinc binds them both with equal affinity throughout the concentrations. Obviously, then the question becomes, what is arsenic doing in terms of M6A in long-term cultures, long-term exposures? So we went back to our HECAT cell line model and looked at the 3 time points that we have talked about earlier, 7 weeks, 19 weeks, and 28 weeks. And very surprisingly, we found that at 7 weeks, there was more M6A, total M6A in the cell compared to passage match controls. However, that soon was suppressed by 19 weeks when cells are starting to transform. and was further suppressed when the cells have completely transformed. The next question was, can this be explained by the expression status of the writers and the erasers? So we did that, and we did see some changes, but there was no congruence between how the writers and erasers were getting expressed versus what this pattern was. In fact, if you look at 28 weeks, when there is the least amount of M6A, we see almost all the readers are induced, which suggests that this is a homeostatic response by the cell to combat the plummeting M6A levels in the cell rather than explaining why the cells have this. Obviously, the question now is, what are the targets that are differentially methylated that could contribute to carcinogenesis? And while we're working on it currently, we have some indications from our already published gene expression data. So I did a literature search and came up with a list of 80 genes whose differential methylation leads to differential expression and contributes to carcinogenesis, of which we found that 12 were differentially expressed in our arsenic transformed HECAT cells, as shown in B. Interestingly, 7 of them were actually differentially expressed way before the cells actually became transformed. So we think those are probably the most likely contenders for differential methylation. We were also able to show that differential expression of none of these mRNAs can be explained by differential expression of cognate miRNAs that target them. So clearly there is some other mechanism that's going to leading to this differential expression, and we think differential M6A might be one of them. We also showed that differential expression of each of these 12 markers is associated with carcinogenesis across a wide range of tissues, including every single one in skin. So that's already known. The question is how this is skin. Okay. We did a gene ontology analysis, and there's only 12 genes. I didn't expect to get much, but lo and behold, I got around 20 different processes that are enriched. All of them are driving our cancer initiation, progression, and metastasis. String analysis also showed that 10 of the 12 proteins interact extensively among them, driving these processes that are enriched here. So we have shown that arsenide binds to metal 3 zinc fingers, suppressing the M6A methylation in a very zinc-dependent manner. And we think that this could lead to differential methylation and expression of a core group of oncogenes and tumor suppressors, leading to the differential expression of carcinogens. And this really forms the backbone of our one application that's currently under review. However, with all this data, the big question remains, does arsenic zinc prevents the effect of arsenic on cellular disruption of certain processes? Does it do anything to the disease outcome? Does it stop the cells from being transformed? And that was the very question that was raised in this forum when I presented last time 2 years back. And I thought that was a fantastic question. So I wrote and secured an American Cancer Society grant to study exactly that, where we exposed HECATs, which are skin cell lines, and BS2, which are lung cell lines, to 100 nanomolar arsenite, and concomitantly supplemented them with zinc for 20 or 28 weeks. And this work was driven by my research manager, exceptional Ms. Sheila Thomas. And at the end of exposure, we measured transformation by epithelial mesenchymal transition, as well as encouraged independent growth. We were able to show very clearly that in arsenic exposed cells, epithelial mesenchymal transition is happening, as we see a clear loss of epithelial markers and a clear gain of mesenchymal markers across both the cell lines. So this suggests that cells are probably getting transformed. However, cells supplemented with zinc are not undergoing EMT. Next, we did anchorage independent growth, and I was really surprised by these results, how black and white they were, not expected. We see clear colonies growing in just the arsenic exposed, but not one single colony across the 2 cell lines in cells that are supplemented with zinc. So hopefully, I have been able to convince you that we have identified RNA metabolism is a major target of environmental arsenic carcinogenesis. And it disrupts RNA metabolism by targeting a few well-chosen zinc finger mediators. And it does so at 100 nanomolar concentration, which corresponds to what we see in the plasma of people who are exposed to arsenic. So this is more likely that we can extrapolate this mechanism to explain what's going on in exposed populations. And finally, we've identified zinc displacement as a mechanism that probably is driving multi-organ carcinogenesis from environmental exposure. So where do I go from here? So currently I'm working on multiple different projects. The first one is as a part of that currently funded ACS project, I'm working with Dr. Watson, Dr. Marchant, and Dr. Beverly to develop a high throughput redox proteomics-based method that will allow me to simultaneously identify which zinc fingers are being targeted by arsenic. Because let's face it, studying one at a time gets tedious and very labor intensive. So if we can figure out a way, a high throughput way that allows me to identify multiple at a time, it gets that much easier. And then we can apply the same technique to a different wide area of different models. Secondly, I am also studying multiple different processes that are already known to be targeted by arsenic, including cell cycle methylation, mRNA methylation, and differential alternative splicing. And I am working with Dr. Kling, Dr. Yad Danapudi, Dr. Park, and Dr. Jay Wong. And really the long-term goal is to take this zinc supplementation into human trials. However, we don't really have many arsenic-induced skin cancer patients in US. So I, along with Dr. States and Zinpro Corporation, which is a respected name in the space of nutrition, trying to set up a human trial with one of our Bangladeshi collaborators, which will probably take some time, but it probably will be the most important thing I ever do. I have to thank a lot of people, including my excellent lab members, Dr. States for his tutelage mentorship over the years, and all the lab members, many intramural collaborators who I learn from and work with every single day, and my extramural collaborators who bring a lot of expertise to the table that I do not have. Also, I have to thank the funding. So I started most of this published work I have shown using the funds from NIEHS. Both are Dr. States as PI, and I was later OI on them, which helped me to get the faculty position with funding from CIEHS as the career development awardee, as well as the pilot project, and with substantial startup funding and research enhancement grant from JHFE. All this together helped me to secure my first extramural funding from American Cancer Society, which I'm currently working with. So at this point, I will thank all of you for your patience, and I'll be happy to take any questions, criticisms. Yes.
That was a great talk, Mayukh. You covered so much of the really great work that you're doing. I have a question. In you're using multi-omics, I'm curious to know if you have also done metabolomics to look at SAM levels, the universal methyl donor, and components of the methionine cycle.
Well, I would
yourself.
refer Dr. States because he has the purse strings. So.
Okay, I think that would be interesting to look at. Also, have you looked at, another idea is, have you looked at the stability of the C3H1 or C4 zinc fingers that you have looked at, and that would include metal 3 in response to inorganic arsenic in your
The stability of the zinc fingers themselves?
Of the proteins, yeah, of the proteins.
I haven't, but that's a great idea. I mean, that should be pretty easy to
It
do.
would be very interesting, yeah, because you could just do a cyclohexamide experiment.
Yes.
But you might mention the results from when the arsenic-bound peptides sat around.
Oh, yeah. So here's an interesting part that I didn't show today. So we were working with the 0NB2 peptides, and we incubated them with arsenic. And then with zinc, the idea was zinc is going to displace. And when we looked at the results, it was all arsenic bound. And we found that if you leave them long enough, so zinc will displace arsenic first, but if you leave them long enough, arsenic is going to bind again. So it's kind of like a cycle. And I think that is really driven by how much arsenic is in the system, how much zinc is in the system, and what is the affinity of that particular zinc finger.
I did
is
note
going
that
on.
you were using a lot higher concentrations of zinc to displace your 100 nanomolar.
Right,
So
but
it
this
seems
is
like the
still
arsenic.
very well
Yeah.
within what,
Yes.
Our biology, like our plasma arsenic concentration is, plasma zinc concentration is anywhere around 10 to 17.5 micromolar. So we are using very well tolerated levels.
I should let
Yes.
somebody else ask.
A really nice talk, Mayukh. So, okay, so you built up this case about, why these rodent models are not good models for arsenic-induced cancer, and you also talk about the importance of the dose. So you're looking at people, let's say in West Bengal and Bangladesh, are exposed to really much higher levels of arsenic and getting those kinds of skin cancers that you were showing. And then you're also talking about environmental exposures that, me and you and everybody else in this room might be exposed to. So my question, what I'm missing out is there any data out there that show that these environmental exposures that we are exposed to in our, chronically everyday life could cause those kind of skin cancers with time?
Well, not with skin cancer, but I think Natalie Dupree just published a paper where she showed arsenic and a host of other metals, even at regular, concentrations that we don't think are toxic, not even less than 10 parts per billion, is associated with increased, I believe it was breast and prostate cancer, and in a Louisville population, breast and colon, yeah. So, yeah, there needs to be more studies, I agree. And even if we do not see cancers, we do see a lot of other effects, like we haven't talked about the lifestyle associated disorders, muscle wasting. Those happen at much lower, and there are good animal models for those.
All right, thank you. And then I guess my next question before I lose my train of thought is, so with the skin model, it's a great alternative model. We use rodents all the time, and it's very nice to see that there are other NOMs out there. But when we're talking again about, arsenic exposure in these different populations, there's a lot of factors that play a role, like genetics and so on. So how, I guess, significant and relevant is the skin model to those special populations, important populations that are actually really exposed to these, high levels of arsenic?
Yeah, so if you look at, the genetic susceptibility, which I did my PhD on, there are multiple very low intensity genetics polymorphisms that are affecting it. But I don't think that really drives the carcinogenesis per se, but modulates the risk factor a little bit. So I don't think that this is a bad model to study those. I mean, every model is bad, some are useful. So, this is what we have right now. But I think one thing that has come up in the last few years is Dr. Stiblo's group at UNC has developed a mouse model which has the human AS3MT gene, and it's showing a lot, not skin cancer yet, but they're showing a lot of population-relevant non-cancer effects at human-relevant arsenic exposures. So maybe we can develop a model that will, help us manipulate a lot of these genes and whatnot and help answer the questions. Does that kind of answer your question?
Yes, thank you.
So for cardiovascular disease, the dose response curve in epi studies goes right down to 0. So even very low exposures enhance cardiovascular diseases in exposed populations. Then trying to remember the other, in terms of the skin cancers in animal models, there's a fundamental physiological problem, which is that human skin and animal skin is fundamentally different, right? So, and you don't have the same sort of progression from the basal layers to the other layers in animal skin. I think that's the real reason why it hasn't worked at all for skin cancer. We're currently thinking that it's not zinc deficiency that may be the problem, but there's something called zinc insufficiency. So you have some zinc, but just not quite enough, right? And in the diets in many parts of the world, that's probably the case because they don't eat very many animal products.
Yeah.
Great talk, and I love the work and where it's going. But I have you may have covered this, but the idea that arsenic displacing zinc and the splicing factors, TRA, 2B and,
No, 0 and B2 and SRSF7. TRA2B are downstream targets of those.
okay. Well, the one that you showed, so the idea here is that arsenic binding is going to change isoform selection due to changing function of
Right.
those proteins, and that you show that there is an increase in selecting of one isoform, increase expression. Is there, if you look at the cancer databases, do you see isoform selection in skin cancer or other disease progressions or in EMT? And you,
Okay, so there are two parts of this answer. In most of these, they're like, with respect to particularly SRSF proteins, right? Most of their expression is kept at a very, very minimal level because going up causes problem, going down causes problem. So most of these, what you see are selecting for an isoform that does not give rise to protein to keep the protein giving isoform at a very basal level. So what this arsenic is doing is it's flipping that and making more of the protein that actually gives protein. So it's just the canonical versus no protein. However,
okay, so you're just, the isoform selection is then promoting increased protein. It's not that you're having.
yeah.
Protein
Well,
isoform
in this
expression
particular
differences
case.
there.
But also there are some previous studies that actually show that certain isoforms are cancer giving, not necessarily SRSF and not necessarily from arsenic, for example. I think it's the INK4 locus, so P16 and P14. One is oncosuppressive, the other is oncogenic. So the balance between the two changes in most cancers. So that is known. However, if you look at all databases, there is really very, very little data about isoforms, which is why we cannot even do that with proteomics because there's nothing to compare it to.
Excellent talk and a lot of the work. I want to follow the Chris question for the arsenic causes cardiovascular disease. And do you think that is also affected by the protein using the arsenic replace that? Is that the target is involved the cardiovascular disease?
I mean, it's possible and it's a very testable hypothesis. I haven't done that, so I cannot say definitively yes, but I would be surprised if nothing was involved.
Okay, so that is a very, very good point, and probably we can collaboration for the
Yes.
cardiovascular part.
Yes, because I know
And,
you already did that with Dr. State and RT.
Sure, yeah. For the cancer, and Chris mentioned about the animal model and with human, that difference like scar. And I think for human and that skin cancer probably also affected by the UV and mouse model in the lab and not the UV affected. So that probably arsenic is not directly induce skin cancer, is enhance the UV. No? Okay, you are the expert, so. Okay.
Arsenic does not have a mutation spectrum at all.
Okay.
Right.
Yeah. Why I think about that way kind of is a co-factor because I use the cadmium treatment cannot induce liver cancer. Even a lot of signaling pathway look like is a stimulate.
Yes.
Mhm.
Almost always in areas that are covered.
Yeah.
Like,
Okay.
think of all the photographs I've shown you, it's groin,
Okay.
like
Yeah.
that.
Okay, I take that. But I mean, it's like a cadmium and also is called like carcinogenic metals. And in the animal model, it's not that easy to use cadmium to induce directly induce cancers. It's special in some like strain of the mice or rat. But when I use a C57 and I keep that 40 weeks and 50 weeks, still not induce any cancer yet, but signaling pathway probably change. But when I give the chemical, we call the compound DNA or some HCC diet, the pre-cadmium treatment significantly increase the rate and the frequency. So that means synergy induced HCC in the animal models. So arsenic probably also somehow is interact with some others, not like UV, but from other compound. So the model, is there some like a model you can use a chemical and then give the arsenic to see if that stimulate or not?
Yeah.
Have been done.
Yeah, those are,
It's, yeah, if you, that model, yeah. Okay. I see.
this is what has been done. So you are, for skin, for example, you are using an egregious dose of arsenic and you're using UV, which by itself will cause skin cancer. And even after that dose, arsenic increases what, by 20% maybe, and types of skin cancers that we do not see in humans. So I don't really know how much you can extrapolate that conclusions to explain what's happening in people. Like, I know a lot of groups are working on arsenic and UV exposure, but we do not see any arsenic into skin cancers in the UV exposed, sun exposed areas of the body.
Yeah, I
There's
see.
very different mechanisms.
Because I'm not like a work in the cancer field, but it just kind of is a question. One more question. Sure. And so you are like using arsenic to replace the several some new target. But I know like cadmium and arsenic also replace zinc from P53, right?
Yes.
P53 is major like
Yes.
tumor pathway. And did you see that zinc can replace that arsenic from
Alex is
P53?
working on that. My colleague Alex is working.
Oh, okay. So I'm work on that. Yeah, because that probably is the first idea
Yes.
to check and
We
then
did
go
it
to the
here.
new one. Yeah.
So Alex actually published data that shows that arsenite, she did not do the direct interaction study, but she showed that effect of like the function of P53 BRCA1
Okay. Yeah.
are all down when you expose cells either to arsenic or to cadmium.
Yeah. So in that case, probably she was lucky. She found
Yeah,
the
and she is
pathway.
currently doing because she works on DNA repair. I do not really want to get into that.
Sure. Sure. Thank you.
No online questions? Anybody else? Thank you.